Yeah.
I am Gretchen Gano. I'm Director of Research for Prescott College. I am here to round out the spring of 2022 with our last Meet the Faculty speaker series. This is designed to feature the work, research, and praxis of Prescott College faculty for the benefit of the campus community and to engage external and internal audiences of prospective students, colleagues, and collaborators. Today, I'm very excited to bring you Jeremy Johnson, who will give the talk, Blending Genetics and Phenomics to Combat a Non-Native Forest Disease and a Changing Climate. Dr. Johnson is a forest ecologist and geneticist with broad research interests geared towards understanding the spatial and temporal patterns of how forests respond to multiple threats associated with global ecological change. He does this by specializing in the use of geographic, landscape, ecological, and genetic tools and theory, with the goal of ensuring the long-term survival of forests with effective conservation interventions and management. And without further ado, Jeremy, both virtual and live.
Yeah. Thank you, Gretchen. So you know how many people we have virtually?
About six.
Six, okay.
I'm gonna log in and kind of see who's there.
All right, we have a lot
Yeah.
of people here. Okay, well, this is cool.
Yeah.
This is fun because we get to do this in person and virtually. So I do want to talk a bit about what I've been working on really over the past 6 and 7 years, focused on trying to create a model system with a group of other researchers to use emerging technology, genetics, remote sensing approaches, machine learning, computers, and quantitative methods to develop ways that we can protect forests and improve forest health. So I thought my clicker was going to work. Oh, I turn it on. Hold on.
Can you do any picture in picture?
I had it set up. Okay. Keyboard it is. Not even keyboard.
Do you want me to keep trying that, the remote?
There we go. We're good.
Yeah.
All right. Moving forward here. So, we're here in Prescott. Our institution's in Prescott. We're surrounded by the Prescott National Forest. And so I think it's nice to start by thinking about the ways that we relate to forests. Right? Forests are charismatic parts of our landscape. We enjoy recreating there. Forests, trees in particular, do amazing things. And they're in amazing places. We get to go visit those places. I get to go to those places and do my research. And so this map up here on the left is the geographic distribution of a group of pines in the subsection Strobus. So these are the white pines. Sometimes they're referred to as 5-needled pines. They have 5 needles per fascicle. And in North America and Central America, there are 9 species of these pines, and they range across quite a diversity of habitats. And so a lot of them will grow at high elevations, sky islands, alpine tree lines, and they go all the way down into sort of xeric mixed pine forests in the north. And so that would be something like eastern white pine, Pinus strobus. White bark pine grows at high elevations. They are important economically. So things like eastern white pine, western white pine, sugar pine, use those into production of lots of different goods and services. They're also ecologically important, right? They protect our soils, they provide habitat for wildlife, lots of reasons that we care about these forests. So the USDA released a report a couple years ago looking at assessing the amount of forest that is at risk of vegetative loss as a result of pests and pathogens. Right? And so 33 million hectares of forested land is estimated to be at risk in the next 5 years. That's about 7% of all the forested land. So when we look at this map here, the green is forested land, the red is the areas that are at risk due to pests and pathogens. So that is quite a lot of forested land at risk. We have about 2.5 new introductions of pests into the United States each year. And since the 1600s, there's been 450 pests introduced and about 16 different pathogens. And many of those have gone on to be quite destructive in forests. right? They result in loss of vegetative tissue, leaves, right? This reduces reproductive output, and we have a loss of regeneration in the forests. In the Four Corners region here, you can see there are portions of the forest that are indeed susceptible to these pests and pathogens. Around here, we've got rusts. I'll talk more about rusts. We've got bark beetle, clearly, both Dendroctonus ponderosa, the engraver beetles, and the borer beetles, ips. And aspen is being impacted by oyster shell scale disease. Amber went out there with me and helped map some of that.
Cool is that?
Conifer decline. So there's lots of threats, right, in addition to climate change, which we'll talk about here. Now, if you look at this map on the bottom right that Liebel put out in 2013, it shows the number of pests per county, right? And there's not an even distribution in terms of the number of pests and where you we tend to find that. What do you think that relates to?
Moisture. Not a forest, not a forest.
Yeah, right. So it's correlated with the diversity of species, right? You have higher species richness, you have more pests. What else though?
Time that it's been occupied by Europeans.
Yeah, people, right? You have tropical pressure, you have the movement of goods and services, and the introductions of these pests and pathogens generally come with people and the movement of things around, right?
Also ports.
Yeah. So there's a lot of threats to forests, right? We can think about what's going on in the Prescott National Forest right now. This is the draft 2021 mortality or damage map. It's not released quite yet, but when we look at a portion of the Bradshaw district here, we see in red all the mortality from last year's the beginning of the bark beetle outbreak that had started. It's the largest outbreak since 2004, the 2003, 2004 outbreak. So even around here, we deal with these pests and pathogens. These are not non-native, they're not invasive, right? They've co-evolved with ponderosa pine. But when we have climate change and we have conditions conducive to these outbreaks, they do impact our forests pretty significantly. So the disease that I focus on mostly is white pine blister rust. It's caused by an obligate, biotrophic, macrocyclic fungal pathogen. Macrocyclic just means that it has multiple spore stages. Biotrophic means it needs living tissue to complete its life cycle. It was introduced in a single event around British Columbia in the early 1900s. You can see that on the red line up here on the map. And it spread into the range of the white pines, and it spread pretty quickly from its introduction in the early 1900s through around 1940, really wiping out much of the range of western white pine and sugar pine. And then it continued to spread until recently. It was discovered in the Sacramento Mountains near the Lincoln National Forest in New Mexico in 1990. They backdated that discovery to about the 70s, but it is now in the northern periphery portion of the range of southwestern white pine. Fortunately, it has not yet been found in the core of the species range, which runs broadly through the Sierra Madre and Occidental in Mexico. However, just this last year, a rusted ribes, the alternate host, and I'll talk more about that, was discovered by Wilshire Peak in Southern California. So the disease is still continuing to spread south, and it's really just a matter of time probably until it reaches the core of the range in Mexico. So just to elaborate a little bit on this macrocyclic nature of the rust fungus, I think it's important to think about it because it's got quite complex ecology and life history. In order to infect and kill a tree, it has to go through these different stages. So the first one is that we have basidiospores that come from the alternate host, which is a ribes. That's a currant or a gooseberry. There's lots of species that can serve as the alternate host. That basidiospore will enter the needle stomata and begin to spread through the tissue of the tree. And it creates these necrotic lesions, these needle spots. They're pretty diagnostic. Once you see that, you know that the tree has been infected. So following the infection, the fungal spore, the fungal hypha will spread through the intercellular tissue, eventually reaching the branches into the bole of the tree where it creates these sort of incipient cankers on the tree. You'll start to see orange margins. They'll create a diamond-shaped canker formation. And it's at this point that the fungus then goes through a change and becomes a pycnial spore. And this is the stage where it creates genetic diversity. It will exude these pycnial drops, which is sort of like sap. It's got a high sugar content in it, and so it's sweet and tasty. Insects like it. They fly, they get on it, and they go to close trees. They're also infected, exchange genetic information, and all of a sudden you've got novel genetic diversity in the fungus. Following that, it will, for about a year or two, it will stay in the bowl of the tree and it will start to expand. And the mycelial networks will basically expand vertically and horizontally around the tree. It girdles the tree, disrupts the phloem transport, so there's no nutrients and water being taken up by the plant, and it'll kill the tree. But before it does that, that canker, as it keeps growing, will eventually erupt, which is where the disease gets its name, blister rust, and it will produce these acial spores that they're very powdery, they're very dispersible by the wind. They can go long distances. So given the right context, they're very stable too. So they can go around 1,000 kilometers if the right conditions exist. And then those acial spores will infect the alternate host, the ribes, where they will then change into uridial spores, which are not very stable. They're very fragile. They don't move very far, but they tend to disperse locally on all the ribes, amplifying the disease on the alternate host. Finally, we get to the fall of the season, temperatures begin to drop, humidity goes up, and the spores change again into teleal columns, which is what you actually see on this image right here. And inside the teleal columns, you generate teleal spores that will then reinfect the pines by converting to basidiospores. So it's amazing that this disease spreads at all because it's so complex. It requires distinct climatic conditions, hosts, and alternate hosts, right? But it continues to do so. So in addition to the disease, we have this issue of climate change, right? So you can't even see the blue on this, but we have projections of moving up to 31.5 degrees Celsius. It's getting warmer within the range of southwestern white pine, which is problematic. A colleague of mine, Andrew Shirk, developed the species distribution model here using a consensus approach and a random forest algorithm, showing that for the 2080 projections, using either the 8.5 or the 4.5 concentrations, what we see is that we have massive contraction. All the oranges and yellows are contraction in the range. Black is stable. There's not much in the south. And then we have sort of mixed results in the periphery around here, and then you have expansion up into Colorado. Of course, that is predicated on the ability of the tree to disperse and actually move. So this is really just a climatic envelope, not the distribution that you actually see. Peter.
Just quick clarification. So the 22.9 and 31.5, is that the temperature range of the group, the penis?
Yeah. This is the range of the whole map for the raster, for the projection of the temperatures.
Oh,
Those are the mean annual temperatures.
oh, I see.
So contraction in the range, spreading disease, what does it all mean? A recent study by Rachel Dudney et al. Took some actual data from Sequoia Kings Canyon, and they looked at the movement of the disease, where it's actually moving. And what they found is that the climate optimum is shifting to higher elevations, which is expected in a lot of ecology. We expect movement to higher elevations into higher latitudes. This is problematic for the white pines, though, because so many of them are high elevation trees. They grow in sky islands. They are the high elevation trees. So even if they haven't been exposed to the disease currently, they're about to be exposed because of where the climate is shifting to. So we've got dual threats. We've got climate change. We've got invasive diseases. How do we go about ensuring healthy forests for the future? So my research focuses on trying to untangle these interacting roles of climate change and white pine blister rust. And I do that through a series of approaches based on common gardens where I will grow trees and look at the phenotypes that emerge in different environments. I use remote sensing approaches to try to rapidly identify how plants respond to changes in the environment in an empirical sense. Greenhouse experiments to simulate climate change and how disease resistance holds up under projected climate change, and then using genomics to perform genome-wide association studies to actually look for those genes that are responsible for resistance or adaptation to climate change as well. So this is just sort of a conceptual framework of the way that I do things. I'm going to talk about 4 different projects that I've been working on here that look at how Southwestern white pine, but also a few other white pines interact with climate change and white pine blister rust. And so first, what I'm interested in is what type of genetic resistance actually exists. There's different types of genetic resistance, and I'll talk about what those look like in Southwestern white pine. And I'll use a basic common garden phenotyping approach to do that. Next, identifying the specific location of the genes responsible for resistance to white pine blister rust using genome-wide association studies and a genomic approach. Looking at how climate change will impact the stability of genetic resistance. We use a greenhouse experiment to do that. And then talk about a project that I'm just about to wrap up now, using hyperspectral remote sensing to identify the presence and progression of the disease. So Southwestern white pine, it really has 2 different regions. We've got this disjunct population that is in the Southwest United States, and then the core of the species range that runs through the Sierra Madre Occidental. We collected 404, we chose 440 families. Those are maternal trees. So go out into the woods, identify a tree, collect the cones from them. They're all open pollinated. So all of the trees that we grow, we know the genotype of the maternal component, but we don't know what the pollen contribution is. Those 440 families were then put into 6 different trials. And I use this term MGR to stand for major gene resistance. I'll explain that more here in a moment. And SY is for sow year. Those are the years that we actually sowed the seeds into the trials. So we have trials in 2014, 16, and 17, both major gene and operational sow year trials. And this is the distribution. The colors correspond to the different years that the trials represent. So all of the Mexico families are in the sow year 17 trial, and the periphery population is spread across to all of those. One other point that I need to make is that Southwestern white pine is at the southern end of this sort of species climb, this range of different white pines as they move from Canada down into Mexico. And they actually interact right around here in the San Francisco peaks with limber pine. And it's an introgression zone. So they hybridize with Pinus flexilis in that area. Pinus flexilis is already known to have major gene resistance. So there's some question regarding if Southwestern white pine does have major gene resistance. Is it a result of this introgression, or is it just because they both have that gene? So this is a approximate Bayesian analysis that looks at the divergence times of the 2 species. And what we found was that 18 million years ago, Pinus flexilis diverged from southwest. The two species, Southwestern white pine and Pinus flexilis, diverged, and then the core of the species range in the periphery diverged about 3 million years ago. There's no ongoing gene flow between these 2 populations. So there's asymmetrical northern movement of genes in Southwestern white pine and flexilis, but there's nothing between the core and the periphery of the range. So it begs a lot of questions about what we'll actually find in terms of the resistance patterns. Okay, so the way that we do this, we grow all of the seedlings in these Rayleigh containers for 1 year in an unheated greenhouse. And then after 1 year of growth, they're outplanted to these boxes. Each of these boxes has a family, so that's a single maternal tree in a row, and then 10 families in a box usually. They're randomized across replicates. So this one has 4 replicates in it. Some of them have up to 6. They grow for another year in those boxes before we inoculate them. Then we take forklifts pick those up, put them on trucks, and drive them to an inoculation chamber. So really just a giant shed that you can control the temperature in. So we put them in an inoculation chamber, and we have to do this multiple times. So usually this happens around September when the ribes have the T-leal columns formed on them and they're ready to germinate back into the pines. So we'll take several weekends to do this. But we'll collect leaves. We have a ribes garden where we can grow and infect some of the ribes, but we have the rust crew that is part of the New England Genetic Resource Center, and they will go out into like Eastern Oregon and up into Washington as well and collect infected ribes, bring those back, place these leaves T-leal column side down on these frames, and then we cover them with plastic to control the humidity. And then we also have these little platforms that have auger and some slides on there. We can use those to monitor the density of the spores as they fall from the ribes onto the pines. And then we can use the auger to monitor germination so that we're confident that we are actually infecting the trees. Once we have that all set up, this tube up above on the picture on the left is our fog machine. We lower the temperature and we turn on the fog, and it gets pretty dense in there. But it doesn't take much more than about 12 hours for this to happen. And so we have a rust team, and they will stay there all night long, constantly checking those slides in that auger. When we hit the right density, around 4,000 spores per square centimeter, we pull that plastic off, pull the leaves off, and they're done being infected. Once that's finished, we put them back either into the greenhouse, if it's the major gene resistance trial, or back into the mounds that we call for the operational trials, and then they are continuously monitored for the development of symptoms of the disease. And that is very time consuming and very slow. These boxes will have 100 trees each in them. It takes around 2 hours, 2.5 hours to count all of the spots on the trees, on the needles, count all of the cankers, measure the heights and the diameters. And we want to do that about twice a year. And when you have 3, 4, 5 operational trials going at a time, it takes all year to do this. So it's really expensive and really time consuming. But we get the phenotypes associated with that. So we're looking for the development of those spots and stem symptoms. Okay, this thing is in my way. I'll make it go.
I think you could drag it.
Maybe. There you go.
Keep dragging it forever. All right, so the first question is, what type of genetic resistance exists? The earliest type of genetic resistance that was identified, and this is done in crops a lot, but in trees, is the major gene resistance mechanism. It's a single gene that confers complete resistance, so it has a dominant effect, right? So we could look at that. All right, so if we're thinking about stem symptoms as the phenotype, major gene resistance basically creates a hypersensitive reaction in the needles where it kills off tissue around the stomata where the fungus came in. Because the fungus requires living tissue, it's an obligate biotrophic fungus, it can't spread past that walled off lesion and thus will not develop stem symptoms. They won't get cankers on the tree. So what we expect is that if the trees have the mechanism for major gene resistance, that they should segregate in a Mendelian fashion. I'll show you that here in a moment. But because these are open pollinated, all we know is the seed parents genotype. So we expect a ratio of 1:1, right? If you have 10 trees, if you have a heterozygous major gene, half of those trees ended up with the big R gene and the other half didn't, and you have a ratio of 1:1. And that's in comparison to the others that will all develop stem symptoms and eventually die. So I use an exact binomial test to test for this segregation ratio. It looks at successes and failures. It's sort of like a chi-squared test. But this, you'll recall from high school biology. This is a Punnett square, right? So if we know that the maternal tree is heterozygous for major gene resistance, and we're assuming that the pollen donors have no resistance, our expectation is that we get big R resistant, big R resistant, little r, neither one of those is resistant, hence the 1-to-1 ratio. Okay, so what we found is across the entire range, we identified 40 different families that did in fact segregate for major gene resistance. That was quite exciting because we only thought we were going to find them in the periphery of the range due to the hybridization with limber pine. But they actually occur pretty regularly at a frequency of about 10% of the population throughout the range. I will point out right here that we have very high infection, which is good. In terms of the number of trees that developed spots, which just demonstrates that we were successful in inoculating the trees. Basically, it's 100% for all of those trials. And they also have fairly high percent stem symptoms. So somewhere between 89 and 95%. So this was really interesting to me. And this was the first part of the project that I was able to wrap up. I have a paper that's going to be submitted this summer with this data in it. But the next question that was of interest to me then was, because we know that there's no ongoing gene flow between the periphery and the core of the range, and we do know that there is introgression with limber pine in the periphery, is the major gene the same gene, right? Are they actually the same gene, or is this just a coincidence? And we already know from several of the other white pines that the major genes are not the same, right? Here's an example of it. We've got Western white pine, and I wish this would go away. It's distracted. Can you go away?
Maybe those 3 dots on the right.
Don't touch those.
Don't touch them. All right, I'm going to move it around here. So Happy Camp is in California, and the other one is Cottage Grove, Oregon. Sugar pine occurs around Happy Camp, and Western white pine occurs in Oregon. And what we see here is that those trees that have major gene resistance actually don't hold up in the reciprocal locations, because virulence has evolved in the rust to take out major gene resistance. So in Happy Camp, for instance, if you have major gene resistance, you're just as good as a susceptible tree. You're going to die because the fungus has evolved to overcome it. Same thing with Western white pine in Oregon. Major gene resistance doesn't hold up. However, if you plant them in the other locations, sugar pine does just fine with virulent rust in Oregon, and Western white pine grows just fine in the Happy Camp area. So they're not orthologous, they're different genes. So what we did to test this is we used a genome-wide association study. We sequenced, what I did was I chose a couple of families from the Bradford Canyon area of the Lincoln National Forest. That was where the rust was discovered. It has some of the highest levels of resistance on the landscape because of the strong selection pressure that occurs there. But collected the needles off that and extracted the DNA, and we sent it off and had it sequenced. And then we basically did a fancy correlation analysis to associate the markers, the genetic markers, with actual genetic positions on the pine consensus genome. And the pine consensus genome is from loblolly pine, but it's close enough to get a good idea. We also chose families of limber pine. And what we see here, we looked at some different measures, but on linkage group 8 of the consensus pine genome, both limber pine and southwestern white pine's genes match up almost identically at the same position on the chromosome. So that suggests that they are in fact the same gene. Again, we only tested this in a couple of families in New Mexico. This needs to be tested on the families that we've identified in Mexico to say the same thing.
That's grand.
Yeah, it's coming. But that was really exciting. So we have the actual position of the gene. It's also very useful from a breeding perspective, because now that we know where the gene is, we can develop a very simple SNP chip and do marker associated selection. So not only can we stop growing those trees for 3 years before we even get some information, we can just take the DNA and say, this is where the gene is, it has that type of resistance. That works for simply inherited genes. It's a little bit more complicated when we work with quantitative resistance. Okay, so we've got major gene resistance. We've got the position of the gene on the chromosome. So there's also some anecdotal evidence about how temperature is impacting the stability and durability of these genes in the future. A lot of model organisms don't do well in warmer environments in terms of the way that they interact with the diseases. And we have seen that in Canada, some of the trees that we know have big R resistance for major gene, like every single seedling should be resistant 100% of the time, have started to develop stem symptoms. And it's like, what's going on? Is it genotype environment interaction? What's the problem? So my colleague Richard Snesko and I put in a proposal and got funding through the Gene Conservation Program, the USDA, to set up an experiment to look at 5 different species of white pine that have known resistance and do a greenhouse simulation of climate change to see if we can knock down some of those resistant families. And so we chose western white pine, sugar pine, southwestern limber pine, all of which have major gene resistance. And then we threw in white bark pine, which does not have any known major gene resistance, but has a high level of quantitative resistance. And what I want to focus on here are the homozygous dominant big R families from western white pine and sugar pine, because they're the most interesting here. So we set up 4 different treatments here. We use an ambient raised bed. That's what we normally do at Arena, just when we do our normal screenings. Then we have an unheated greenhouse, a warm greenhouse where we use heating mats to warm underneath the trees. And then we used a wet wall greenhouse that we can cool the temperature down in. And all of the seedlings were then replicated in those treatments, and we have uninoculated controls in there as well. We put in hobos to monitor the temperature and humidity, and we were trying to match about a 4-degree difference in temperature between the cool and the warm treatments, and we got pretty close on that. I'm gonna have to think about it in the future where I position things. I don't think other people can see that. but I
No,
can.
no one else can see that.
Just the live audience. So we did succeed in creating a 4-degree gradient in temperature across these treatments. And so we measured between October 2020 and July of 2021. And so what this figure right here shows you is the number of cankers that were observed at the 3rd inspection by species and treatment and resistance type. So we've got quantitative resistance, heterozygous resistance, homozygous resistance, and then susceptible. And so the ones you want to pay attention to are the western white pine, sugar pine, these blue ones right here, because what we see is that there is in fact canker formation in these homozygous dominant genotypes, and that shouldn't happen. So if I separate that out and I'm only looking at the homozygous dominant families now across the treatments, so ambient, greenhouse ambient, heated and cold, what we actually see is that we have a statistically significant increase in canker formation in the heated treatments compared to the others. So something to consider. Yeah. It means that that type of resistance is not durable, right? In future climate change. We've invested over the last 50 years, the government and taxpayers in programs to breed major gene resistant trees and to put them on the landscape. Millions and millions of dollars have gone into these programs. It might be time to think about a different direction. So what is that different direction? What it says underneath there is another type of genetic resistance. Okay, so
I've learned how to remove it.
how do you remove it?
Well, it's risky, you could blow the whole thing up, right?
No.
Control Alt Shift H.
Oh, don't do it.
Don't
Well,
do it.
Do
I'll try it
it.
at the end.
up, up, down, down, left, right.
Right. I think it's a control.
The secret's in the more.
Okay, well, I'll look at it in a second. So the other type of resistance is quantitative resistance. And what that means is that there's a complex pattern of inheritance. So it's an additive form of resistance where there could be 10, 100, 1,000 genes interacting with environments as well, contributing to a range of resistance. So if you use survival as sort of the ultimate diagnostic phenotype, if you survive, you're resistant. There are other mechanisms associated with it I'll talk about, but we could look at that in the same way. If we were comparing to MGR, our expectation of a segregation pattern from 1 to 1. For quantitative disease resistance, you might see slightly lower survival, or even lower, right? But you can also find higher. And this is all relative to the susceptibles, where 100% of the seedlings will die at the time. So the mechanisms that are generally involved, sort of like the hypersensitive reaction, there can be slow fungal growth. There can be metabolites that slow down the movement of fungus. There could be endophytes involved in needles, so other fungus and bacteria that interact with it and slow it down. You can have what is essentially a hypersensitive-like reaction, but in the stem of the tree, where it creates a bark reaction. So another form of necrotic lesion kills off all the tissue around it, and then the canker can't spread because there's no more tissue for it to consume. So I did a pilot study on this where we looked at 40 families from New Mexico that we had been growing for quite a few years and had some good data on it. And what we see here, so we have the Cibola, Santa Fe, and Lincoln National Forests. And in the Lincoln National Forest, that's that Bradford Canyon area where we selected the trees to do the genome-wide association study from. They're in green. They have the highest level of survival, right? And the mean severity of the infection, which is based on how the cankers expand vertically and horizontally. It's a standard index that the Forest Service uses. But what you see here is that there's actually quite a wide range of distribution in terms of the percent survival across these different families. And even in the Cibola and the Lincoln National Forest, where you don't have nearly the level of survival that you do in Bradford Canyon, you still have 2 families that have really high survival. And this was almost 9 years of following them. After the first few years, most of the damage was done, and those that were alive stayed alive. So some of the correlations in looking at the percent survival as a function of the percent of stem symptoms on the trees, there's a strong relationship between the number of stem symptoms and survival, as well as bark reactions. And so we see completely healed cankers on some of these trees at this point.
Strong negative correlation.
Strong negative correlation between survival. Yeah. So how does that look across the entire range? A lot of these studies that I published recently have just been based on a few sort of case studies in the periphery because the trials are ongoing and we don't have all the data yet. We've started to look at the range-wide patterns of survival and quantitative disease resistance in all 440 families at this point. And what we're finding is very similar and consistent patterns across these different trials. So the cellular 14 dotted lines have major gene resistance. The solid lines are quantitative resistance. And in all cases, we see a distribution that is emerging, suggesting quantitative forms. The next step with this is actually to perform a much larger genome-wide association study and to look for outliers that correlate with these phenotypes. So that's coming up. All right, the last thing, and again, just like with the major gene resistance, when we find outlier loci, we can then use that to develop marker-associated breeding strategies so that we can actually create a program to breed trees that have more durable resistance. We can put those into orchards, collect seeds, and use them in restoration and reforestation. So the last project that I'm going to talk about here is the Special Technology Development Program that I received a grant from the USDA to do. So this was a grant that I got about 4 years ago at this point. And the idea was to develop a new platform that would be able to identify rust in the pines before you could see it, and also improve our ability to track the progression of the disease. Because as I said earlier, it's really expensive and really slow to go out and measure all of those phenotypes. And so the way that the technicians at the Drina Genetic Resource Center rate things, they kind of use this, it's almost subjective, but they're saying that a tree is vigorous, moderately vigorous, sickly, or dead, right? Just by looking at it and saying, because the disease, it's infected with the disease, it's at a particular stage. But if we can create a more quantitative approach to that, it would really be beneficial. It would reduce assessor bias as well. So this project had funding in it to hire a PhD student. So Maria Hagsma was my PhD student who worked on this at Oregon State, and she's got a couple papers out of it now. So we started out in a greenhouse just to pilot it to see what would work. We chose 10 families and we completely randomized them. We had half of them inoculated, half of them were uninoculated controls. And then we engineered what we call the hyper rail. And the hyper rail is totally covered up for the in-person. The hyper rail is an extruded aluminum rail that we mounted on the top of the greenhouse. We 3D printed this housing for the head wall camera. It's a hyperspectral camera. We have a step motor on there and our Arduino board, so we can program the speed that the camera moves across the scene, so we can create equal exposure depending on the amount of light that comes in. But it's automated. It's like a little robot. It basically just drives across the greenhouse. So it's a push broom sensor. It has to move. It's not like a normal camera where you click and shoot. It has to be moving to collect the spectra. And it has about more than 400 different colors essentially that it sees, right? So unlike us, we can see a few colors. It sees a lot. So from near infrared, the visible spectrum, and we can use those to assess the different biochemical properties of a plant. Because of the way that it interacts with light and reflection, that can tell us something about its infection status and health. So we took 16 different camera readings. Those are the arrows up there. And then we had 4 different inspections by the Drina technicians. And we have these 10 families, and they have a range of known resistance types, ranging from completely susceptible up to major gene resistance and high quantitative resistance in there. So what did we find? Well, it was exciting. What we found was that we could identify more than 80% of the seedlings that were infected early on, when the technicians could only see about 20% of them. So it's pretty effective at noticing that there's some stress starting and that there's an infection within the plant. In terms of predicting and classifying the trees into these different categories, in an automated sense, we used a support vector machine learning approach to try to automate the process of categorizing them into these health categories. And we were able to get the sort of lower end of healthy trees pretty good. And we could definitely tell when the trees were dead with the camera. But those areas in the classes in the middle were a little bit harder to match. A lot of that has to do with the subjective nature of the classification and what we were basing that on, comparing to the Drina inspectors. But if we combine those two classes together, we improved our accuracy. But overall, we had close to an 80% accuracy in terms of placing each tree into the correct category. So we can identify the presence of the rust pre-visually, and we can track the progression of the disease at a really high temporal rate compared to what the technician would be able to do. The next approach was, those cameras are really expensive, by the way. Like the Headwall Nano VNIR, very near infrared camera, is pushing $150,000. It's really expensive. And so it's not practical to put on a drone, not really. And it's not practical to just deploy out into the forest and use it to assess the presence of rust. And so the idea is that if we can identify the right combination of bands in that camera that are actually identifying the rust, we could just engineer a simple sensor fairly cheaply and deploy that. And so we looked at all pairwise comparisons of spectra. We used all of the normal, vegetation indices that are out there, like the normalized difference vegetation index. And we ranked them using a new search algorithm that was based on the Bhattacharya coefficient. And what we found here was that the photochemical reflectance index, we used the normalized photochemical reflectance index, was the best index at identifying the rust and placing the trees into their health categories. This index is associated with photosynthetic capacity, light use efficiency, water content. Makes sense as the trees are getting less water from the disease, that we can detect that. So by taking just the photochemical reflectance index and comparing it for infection detection and bigger class separation, we only have very minor loss in capability. So going from 83 to 87, 76 to 80. And when we priced out developing a little sensor that does this, we can build one for about $12.
Makerspace.
Yeah, so $12. Yeah, so you can 3D print this little thing, put the sensors in there with an Arduino board, something to record the image. You can send it on drones, and you can put it in different places. So that's really cool.
Yeah.
That was all in a greenhouse setting. The idea that we could scale this up to a real forest or to a location that's not in a greenhouse and under very controlled settings is important, because these trees have water and fertilizer and everything else. And so what we were detecting was the stress induced by the rust, but that's not always the case in the real world, right? So the next step was to scale that up. And what we did was we built the 8-meter hyperrail. We brought it to the Genomics Genetic Resource Center, and the 8-meter rail is, I'll play this while I talk about it. So the 8-meter rail is designed with, I know you can see the wheels on here. We actually put little wheelchair motors on. And so the camera will move down the hyperrail across the reps, recording the images, and then it will slide down and then do it again and slide down and do it again. And so with the STDP funds, we were able to build this outdoor rail and compare it with Alberta limber pine, southwestern white pine. This is, let's see, this is the limber pine. We also did white bark pine, western white pine, a lot of the different species, because the idea is also, if we can just detect it in southwestern white pine, will it be the same signal in the other species as well? So we're still working on that. Here in the last year, we've got all of our data collected, and we'll start trying to piece that story together. We also built a 3-meter rail, portable, the mini rail, and we have begun to take this. This is at Crater Lake, so we took it to Crater Lake. We have a restoration planting out front of the Rim Village Visitor Center that has white bark pine. They have a high hazard there. There's a lot of rust around, and so a lot of these trees are infected. And so, in order to try it in the field, we're taking it there. We've taken it to the BLM Tyrell Seed Orchard as well. But ultimately, those places are all still a little bit controlled. We want to be able to put this onto a drone or something else and look at forest stand level patterns. All right, so I'm going to wrap up here real quick. Just a couple minutes here. I just want to say real quickly some of the directions that I'm moving forward with. I was able to submit a proposal with some of my colleagues about 2 weeks ago to the NSF for the biodiversity and changing climate call. And the idea is to take what we've developed over the last several years with these common gardens, where we have different temperature treatments at the Sega sites. We've got our disease trials that I've been showing you. We've got all of our sequences. We've sequenced every one of those trees using RAD-seq approach. And we're introducing the idea of endophytes into the proposal. So we're taking foliar tissue and we're going to do metagenomics. And we're going to look at all of the bacteria. It's like the microbiome of the plant, basically. And we're going to see if there's a correlation between disease resistance and diversity of endophytes in the needles. And we're going to take all of that information and create a forward in time demogenetic model that we can use to predict resistance and adaptive traits to climate change. And ultimately, we'll put those into permanent field trials, different permanent plots that we can monitor for the future and eventually get those seeds into the landscape. And so all of this, all of what I've been talking about takes high throughput technology and phenotyping, high throughput genomic approaches, machine learning to develop a system that can be transferred and used to rapidly address emerging pests, pathogens on the landscape. Because, like I said, 2.5 million introductions each year. Things like emerald ash borer are ripping across the country, destroying all the ash trees. It can be quite a challenge to deal with, and you have to be able to respond quickly to some of those things. Additionally, we have to get those seeds onto the landscape. And so that involves this first part, which I've been talking about, getting the seeds, testing them. But once we have identified a tree, we need to take those seeds or those trees and graft them, put them into clone banks. We need to protect the actual tree that's on the landscape, that's already there. And then put in durability trials so that we can monitor the progression of the disease in a real landscape. And we've been able to put in 2 durability trials so far. This last summer, we collaborated with the Mescalero Apache Reservation and we put in a durability trial in New Mexico there. It actually flooded with the monsoons and just destroyed everything. So we had to put it back in. So we've replanted. Flooding is a problem,
Yeah.
but it's also a perfect site for infection because of the moisture level. We also have a durability trial here in Eastern Arizona that's doing much better. And, we involve a lot of folks in these projects. And so getting stakeholders involved with the planting, it helps to communicate the science, helps to get the plants in the ground. And it just ensures that we can help communicate the science to more people. So this is an example from the Mescalero Apache planting. We've also created a progeny clone bank with the Bureau of Land Management. This one's in Oregon. We've taken all of our elite trees that we've identified so far in the southwestern red pine trial, grafted them on the side, put them into the clone bank so we can get our seeds and use in restoration. So ultimately, this whole process is to get the trees, identify the resistance and the adaptive components to climate change, and then get those trees on the landscape so that we can have healthier forests. Okay.
Yay. For the good of the online audience, can you have a camera? Can they see you, Jeremy?
I think it's, oh,
I'm not
you want me to do this?
sure.
Oh,
They can see us.
we have the wrong camera on.
Uh-oh. The whole time? Oh, well. No, this is what they should have seen.
Yeah, but now they should be able to see you. So at this point, we normally take questions. And so we have people here in the room and we also have people in the Zoom land. So please, if you are joining us remotely, we're a small enough group, you could unmute yourself and ask, or if you're shy, you could type something into the chat and we will have Jeremy address that. Questions?
I have a question.
Yeah, of course.
So at one point you said that, we've been spending these millions of dollars on these reforestation projects. That was not the right approach. Then I feel like you just ended by saying we need to do more reforestation, but I'm missing something.
No, but we can't be focusing on major gene resistance. What I was saying is most of the last 50 years and all the money that has gone into these breeding programs for resistance has been focused on major gene resistance.
Right.
And breeding for these single genes, but they're not durable because virulence arises and the interaction with climate change could knock down that single gene. That's not true with quantitative approach, quantitative genetics, because it's hard for the fungus to overcome 100 genes, to evolve virulence to more genes like that.
Okay, cool. Thanks for that.
And there's more sort of mechanisms that are involved in that type of resistance too. So it's not just bark reactions, for example. It could be a lot of other things in there.
Didn't one of your graphs sort of just a few back where you were comparing the dotted lines, which I think was the quantitative, and the single solid lines, which was the MGRs?
This one?
It was further down maybe, but where you were just comparing the two, you had a bunch of images. Yeah, that one,
This one?
right. I thought I saw that the solid lines. I thought I saw that the dotted lines had less severity and greater survival. Okay.
No, yeah, so we have average severity.
Oh, so the
they have lower
MGRs, that's my
severity,
confusion.
and they have higher survival.
So don't the it seems like the MGRs have higher
They
survival.
do.
So then I'm confused with your answer.
But that's just because they're in a very controlled setting. There's no virulence. Yeah, but in the landscape where virulence arises in the disease, that's what this other figure is. So this paper that I published on Western white pine and sugar pine. So what this shows is percent with stem systems. So if we're looking at a happy camp here, we've got two different, we've got Western white pine and
Right.
sugar pine.
Right.
The percent with stem symptoms for sugar pine is way high. They shouldn't have any of those stem symptoms,
Mhm.
but it's because of the virulence.
We're looking at the dotted black
Yeah, the
lines.
dotted black lines.
Right.
Yeah. So, and because they're different genes, right? So it's knocked out basically MGR in sugar pine at one location, and it's knocked out the Western white pine in Oregon.
Mhm.
So not,
It looks like Lori is saying thank you, and Muriel has typed in a question, but Muriel, if you want to unmute, you'd be welcome to do that. You seeing that,
Yeah.
Chairman?
so Mariel's asking, what you see is the window of opportunity for understanding and acting in the case of the pines. What is the necessary time frame and amount of seeding to preserve enough of the forest structure for the medium tenure? I mean, I think that's a great question. Trees, unlike agricultural crops, have to be on the landscape for hundreds of years, right? So we have to have that durable resistance. You know, in the short term, short to medium term, you really have to focus on those areas that are high hazard, where there's a lot of rust already present, and try to get the resistant seedlings into those stands first so that the genes can be swept actually to a higher frequency, and then focus on the lower hazard sites later on. But yeah, it's a challenge for sure.
Thanks. I kept trying to unmute and I couldn't remember how to do it.
Thanks, Muriel.
Yeah.
Are other people studying the intermediates, the vulnerability of the intermediates or fungal pathogens for the fungus or?
I mean, like the alternate host, I mean,
Yeah,
Yeah.
or attacking the fungus itself with another fungus or something like that.
so the rabies question, people have looked at it. It's a little bit strange. There's resistance in the rabies as well. It has to do with leaf size and chemicals. There's a lot of sort of different nuances there. They're deciduous. They lose their leaves in the wintertime, so they don't stay infected, and the rust doesn't impact them in the same way that it impacts the tree. It doesn't kill them. I mean, they can get really infected and have a defoliating event, but they're going to get new leaves the next year in most cases.
Right.
So it's been harder to deal with. The original strategy for dealing with blister rust was rabies removal.
Right.
And that was clearly not possible. I mean, there's just so much. You just throw money into a hole.
Sure.
There wasn't no hope. As far as the rust itself goes, it's really complicated to work with because of the macrocyclic nature of it. So people can't even propagate it in the lab very easily.
Wow.
So it's been really very challenging to deal with understanding the rust. There's some genomic work on it, but it's minimal.
And I assume you've gone back to its original location and or somebody has
In
it
Asia.
and figured out what keeps it at bay over there.
Well, I mean, in Asia where it's come from, and there are pines there too that it does attack, of course, but they've co-evolved together and they
Pines
sort of have,
resistant.
yeah, they have the little cat and mouse game that goes on, just we have bark beetle here, and it doesn't tend to be catastrophic. in
Can
most
you
cases.
can you bring over genetics from Asian pine and,
Introgress it.
yeah,
You could, yes. You couldn't put those trees into the forest because the Forest Service right now has a no
oh,
genome
sure, they wouldn't look at that.
policy. Because that's just like the problem.
But if there is no forest, maybe then you reconsider.
I know, but that's already true with
Yeah.
chestnut,
Right,
for example. They've got the chestnut, American Chestnut Foundation. They've engineered a resistant tree,
wow,
but they can't do anything with it yet because they can't put it on the landscape. And even private landowners won't do it too because of the chain of command. The primary certifier of wood products comes out of Europe and they allow no GMOs. So if you want to have a certified forest product, you're not going to have anything to do with it at this point. So the GMO approach, though a useful one, could be very useful, we're not to the point where we can actually use any of it.
Gotcha.
Interesting.
I'm calling in from Hong Kong. And sorry, we're still in face masks here. I can take mine off. I'm in my office. Sorry, it's early in the morning for me. I came in to hear this before my day started. I run a program here in Hong Kong. Well, first off, I'm from Montana. That's my US based home. And I've seen the the devastation when I'm home in the summers to the forests, to, large swatches of Yellowstone National Park. And I've just watched those acreages of brown trees go through the landscape and, stunned by what's possible and what isn't possible. But here in Hong Kong, I have a molecular biology lab for my high school students. And we do a lot of ecology-based genome sequences. So I have capacity to do both long reads and short reads of various genomes. And we have focused on, and I'm this may be off topic a little bit, but I just want to understand if there's a little bit of a connection. We do a lot of soil-based ecology, bacteria, but we've been looking at these sequences, and I don't know, I think we've sequenced 26 genes, this, bacteria in the last academic year. So we're pretty productive with our kids. And we've got a whole lot of high school kids doing this, but are you finding that there are parts of the ecosystem? I have to believe that there's something in the soil that might protect these trees or these shrubs or their root systems. And that's kind of the journey that we're looking for. And we found some, I guess I would call them helpful bacteria that support our forests here in Hong Kong.
Yeah, I mean, it's definitely true. And we're trying to do the endophyte study. We haven't done soil analysis in the mycorrhizal associations and that kind of work yet. I think it's also important, but it's sort of the same approach. Right? So what we would do with the endophytes in the needles is basically do a metagenomic approach where we just try to identify all of the bacteria that are present and then see if there's a correlation with the types of resistance. But we already know that it's true in some plants and in the soil in particular. And again, with fungal associations, there's some really good evidence about how it contributes to disease resistance. Like the classic example right now in the Southwest is the work that Kitty Gehring has done with Tom Kolb and Uvia Flores Renteria out of San Diego State. And they looked at pinyon pine that had two genotypes. One was drought tolerant and one was resistant to a moth defoliator. And when they looked at the mycorrhizal associations, they had two different and distinct associations. And that helped to confer the actual resistance and also water uptake in improving water sufficiency. So it's really a relationship.
So we're not set up to do any of the fungal studies. I mean, we've kind of had to just make, simple bacteria our goal, really. But we have found some interesting, the soils here in Hong Kong are so poor that, and yet that we're, reasonably, we have a lot of country parks, so there's a lot of forest around us. I don't know if you can see behind my, I think I have it on blur, but we're surrounded on three sides here at the school by country forest, but country park forest. But the soils are so poor here that we've actually had to come, the plants have adapted to this. Would be part of it. And I'm just curious where the helpers are.
Yeah, I'm curious too. I mean, I'd like to do that kind of work as well.
I'll send you a paper.
Cool. I appreciate it.
Yeah.
Thank you.
Nice to see you, Diana. Bye. Are there other questions?
I'm gonna have to leave. Take care.
I would just like to ask one.
Sure.
My name is Otto Schwartz, and we're in Eastern Tennessee. And I've worked with Eastern White Pine, and not to the extent, the broad extent that you have, and education that you have, but I'm a physiologist, or was, I guess I still am, physiologist and biochemist. And I worked at cloning Eastern White Pine to
Okay.
try to find individuals that were resistant to various environmental stresses. And all of the environment things that are happening with warming and so forth brings up a question that I don't know how to answer, and I'm not sure you do either, and I'm not sure that it's appropriate here, but I'm going to ask it anyway. You said that your trees would be out in the field for what, 50 plus years?
Yeah, easily.
Okay. Do you have any idea of what the climate temperature range will be out 50 to, well, let's say 75 to 100 years? See,
Yeah.
that's what bothers me about this. Not that it nullifies anything you're doing, but I don't know how you gauge for responses both on the tree part and on the fungal part.
That's a great, I mean, it's true. So what we've done to understand climate change is that we have 3 common gardens. I didn't talk about any of them here, but we've planted 3 gardens at very warm sites to try to identify and understand what the adaptive components of the climate and environment are. And then we've done the disease stuff. And we're trying to combine that to better understand those relationships. The truth is, when we brought up many of these families and we plant them at warm environments, they just die. Right? I mean, they actually don't do very well in the warm environments. So identifying resistance is one thing. If climate change proceeds rapidly and we don't do something like assisted migration, then I mean, they're going to suffer tremendously. There will be no regeneration, and then I guess it's a moot point. But yeah, there's a lot of interactions, and that's what we're trying to link all of this together. And some of that has to do with the modeling approaches that we're proposing moving forward to make predictions. But yeah, without good phenotypes and having good systems to understand what those phenotypes are in the changing environments, you're going to have a really hard time.
Well, I'm sorry to bring that up, but it concerns me, not just, I'm not a forester, but just as a concerned individual, that what is the resilience of the general genomic population to temperature change? And that's kind of what I've asked, but.
Yeah, we're working on some of the genome-wide associations with the temperature in the climate gardens right now. I don't know what they all look like at the moment. We'll see.
Well, listen, thank you. I appreciate your time.
Yeah, absolutely. Peter.
You talked about testing it in the greenhouse with different temperatures,
Yeah.
but it's also predicted that it'll become drier as well as hotter.
Yeah.
And the fungus, it seems, needs humidity. Is it possible that the tree's humidity tolerance or drought tolerance could save it from a less drought tolerant fungus?
Yeah. So, well, potentially. The study by Duggan et al., if you read this paper, what they showed was, let me see if I can. I'm just going to delete this. It's easy to see. They were looking at the distribution of the pines, but also the ribes and the interaction with temperature and where it's all moving. So some of the, as it moves up, you have fewer ribes at higher elevations, but the temperatures are conducive,
More conducive.
and you have higher humidity. So those trees that are down here are dealing with less pressure from rust, but they're dealing with higher temperatures and reduced moisture precipitation. So that interaction is super complex. I mean, that's really hard to pull out. You know, the stuff that we've done in the greenhouse with simulating the climate change, that was just to knock out that gene. However, we also in the greenhouse with the hyperrail and the inoculation have been looking at, we have drought treatments as well. So we have a 50% water reduction and
So you
100%
were-
water
okay,
reduction in addition to the rust to see how that interacts.
Right.
But it's true, and it's been noted for a long time. The climatic conditions of the fungus and the way that it has to have these particular situations to progress through its life. It suggests that it wouldn't spread into Mexico. Like you're not going to get that,
I was
but it
wondering.
keeps on moving south anyhow. But I
And the winds have to be going that direction
Yeah.
also.
yeah, or people have
Or
to-
people be carrying it.
So when I asked you what question you would want to be asked, I think it involved something about how do you translate this
Yeah,
approach to other settings, other species, and a reflection is just that it's there's so many methodological things that you have in the cookbook here. Yeah.
it's true. And the problem, the big bottleneck for most of the history of forest breeding is the time and the phenotyping, right? Trees grow slow, you know, and if you want genetic gain on anything, you have to like do these things very, very slowly. And the only way to improve that is to speed up the breeding process. And the only way to do that is genetic engineering
Of the breeding process of the trees or of the
Yeah.
fungus?
of the trees, like
Of
to
the trees.
improve the trees for both climate resilience and disease
Yeah.
resistance.
Oh, right, because their generation time is
Yeah, because
so slow compared to fungus.
exactly right. And you have to anticipate, as Otto said, you have to anticipate 50 to 100 years, the climate then.
Right.
That's your target. It's not now.
Yeah,
Right.
So
yeah.
And that's just one of the hundreds of pathogens.
Oh, yeah. There's
Muriel
so
saying
many.
too, she was wondering about the translation.
Yeah, I think the trick here is what we've done as a system. The Southwest white pine itself, and white pine blister rust is a destructive disease. But the goal is that the approach serves as a model so that we can, as the next disease comes, how do we rapidly identify resistance in trees and rapidly get that resistance into the landscape? And rapid is relative
Yeah.
because again, it's trees. But it should be translatable using the remote sensing approach and the genomic approach combined to merge those things together to speed up the process. That's what I have a good deal of projects that are moving forward, so we'll see.
Sounds like it. Are there other questions from Zoom? As we. Great work, Jeremy.
Thanks, Mariel.
I guess I have kind of the science policy question.
Yeah.
It's if, let's say this works, what are the other ways and organizations and institutions that would need to be mobilized in order to deploy this?
Yeah, well, I mean,
approach?
I think the largest agency that would be the Forest Service, and the Department of, because you have a lot of these species basically protected refuge in national parks, like whitebark pine, Yellowstone, Sequoia Kings Canyon, a lot of parks.
Crater Lake?
Yeah, Crater Lake.
Yeah.
Right. So having those two agencies involved is the big way. And
Yeah.
that's where most of those sites are at this point.
Fortunately, they move on a dime.
Yeah.
But I mean,
Yes,
do you see, would that need to be like a whole new effort, or
It could.
is it something that?
No, so the Dorena Genetic Resource Center where I did my postdoc and I continue to collaborate with them on these projects. That's a Forest Service facility. It's there designed specifically to do one
To do
thing.
this, yeah.
Yeah, to identify trees. And they do it old school forest geneticist way, quantitative genetics.
Mhm.
They don't have molecular labs and they're phenotypically evaluating the trees.
Mhm.
And what I've done is brought in some of these new tools to their facility. They're using them now.
That's good.
Yeah,
So
yeah,
that's we've been successful because of the ability to get some of the grants and collaborate with them because they're not doing that. They just have funding, right? They
Yeah.
have a long-term funding from the government, which is helpful. Our problem is that we run on a 4 to 5 year grant cycle.
Right.
Yeah.
The NSF and others, I mean, they're not really interested in having a 50-year long funding.
You used to have the long-term,
They have the LTE, I mean, they do have some of those things, and, you know, with Neon and other projects, they have some big long-term commitments.
Yeah.
And they had a new one, we sent a proposal in a year ago or so, it was not funded, to develop a center, essentially, for understanding disease, forest health, basically.
Yeah.
Forest health.
Yeah, you need like, yeah.
And it was gonna be in collaboration with Ohio State, the Forest Service, in some various places, but
Yeah.
there's no other way. So the Forest Service has the commitment in that they have pretty much guaranteed funding
Mhm.
to deal with forestry. But they don't have the mechanisms of advancing technology very fast.
Yeah.
So if you can collaborate with universities and the agencies, then hopefully we can get something put together.
Let's do it. Thanks to everyone
Yeah, thanks, Tony.
for your questions and for joining. And hi, Tony. We haven't heard from you, but it's nice to see you. And Ashira.
All right, well, that's fun.
I think we will be wrapping it up for today, and please come back in the fall when we start again.
thanks for doing this. Kenton.
Yeah.
When are you gonna come do yours on ants?